Have you ever wondered how the human eye develops from a simple outgrowth of the brain into a highly specialized organ for vision? In this video, we'll break down the embryology of the eye in a clear and structured way. The eye is a complex sensory organ that develops from three embryonic sources. First, the neuroectoderm of the forebrain, which gives rise to the retina, optic nerve, and ciliary body epithelium. Second, the surface ectoderm, which gives rise to the lens and corneal epithelium. Third, the mesoderm and neural crest cells, which give rise to the corneal stroma, sclera, choroid, and extraocular muscles. Eye development begins around week 3 of gestation and continues into the postnatal period. The formation of the optic vesicle begins around day 22. Lateral evaginations from the diencephalon form the optic grooves. These evaginations enlarge to form the optic vesicles. The optic vesicle then contacts the surface ectoderm, inducing lens formation. The surface ectoderm thickens to form the lens placode. The lens placode invaginates to form the lens pit and then pinches off to form the lens vesicle. The lens vesicle loses contact with the surface ectoderm and becomes the primordial lens. The optic vesicle invaginates to form a double-layered optic cup. The inner layer of the optic cup gives rise to the neural retina, including photoreceptors, bipolar cells, ganglion cells whose axons form the optic nerve and other retinal neuronal layers. The outer layer gives rise to the retinal pigment epithelium. This invagination also creates the choroid fissure, which allows the hyaloid artery to enter. The retina develops from the two layers of the optic cup. The outer layer forms the retinal pigment epithelium. The inner layer forms the neural retina, containing photoreceptors, bipolar cells, ganglion cells, and Müller cells. Ganglion cell axons converge to form the optic nerve, which becomes myelinated after birth. Primary lens fibers elongate to fill the lumen of the lens vesicle. Secondary lens fibers continue to form throughout life. The lens remains avascular. Nutrition is provided by the hyaloid artery during development and by aqueous and vitreous humor after birth. The cornea develops from 3 layers. Surface ectoderm gives rise to the corneal epithelium. Neural crest mesenchyme gives rise to the stroma and endothelium. Mesoderm contributes to stromal elements. A cleft forms between the lens and cornea, creating the anterior chamber. Mesenchyme differentiates into the iris stroma, ciliary body, and trabecular meshwork. The iris epithelium derives from the optic cup. The iris stroma derives from neural crest cells. The sphincter and dilator pupillae muscles are neuroectodermal, making them rare examples of smooth muscle derived from ectoderm. Eyelids form from folds of surface ectoderm with a mesenchymal core. They fuse temporarily around week 8 and reopen around week 26. The lacrimal gland develops from ectodermal buds in the superior conjunctival fornix. The nasolacrimal duct forms from a solid ectodermal cord that later canalizes. Extraocular muscles are derived from preotic semidomeres of mesoderm. They are innervated by cranial nerves 3, 4, and 6. The hyaloid artery supplies the developing lens and retina. It regresses before birth, leaving the central artery of the retina. Some important clinical correlations include coloboma due to failure of choroid fissure closure, anophthalmia, which is the absence of the eye due to early developmental failure, microphthalmia, a small eye often associated with infections such as CMV or toxoplasmosis, persistent hyperplastic primary vitreous due to failure of hyaloid artery regression, and congenital cataract, often due to maternal infections such as rubella. In summary, eye development is a coordinated process involving neuroectoderm, surface ectoderm, and neural crest mesenchyme. Disruptions at any stage can lead to significant congenital anomalies.
If you found this helpful, make sure to like, share, and subscribe, and I'll see you in the next video.